Thank you, Sandra. So we have already gone through the section 4 of the scientific guidance about the evaluation criteria. So now we will go to the 5th section and last section of the guidance, which is about the technical dossier, and it contains subsections about the recycling process, the decontamination process, and the post-processing. Then you will hear about the determination of decontamination efficiency, and then we'll have the self-evaluation in the guidance. What I will try to explain to you is what we need to have in the technical dossier. So I hope this will be useful to the various potential applicants that may be in the room, but also you will understand how we work, what kind of discussions and questions our working group experts try to answer to when we have our meetings. And when we decide that the technical dossier is really complete and we can proceed, move on with finalization of the risk assessment. Okay. So I hope you will find it interesting. You have already heard that the regulation 2022-1616, which replaced the old recycling regulation 282 of 2008, has introduced some changes. One important change, as Bastian said earlier, is that we set the intricate knowledge of the decontamination process as a basis for someone to become the applicant. So for this reason, the applicant is a developer as it is defined in Article 17, Paragraph 1. Okay, so now we will talk about the three steps of the recycling process. So in the beginning, we have the collection and pre-processing, then the decontamination, and then the post-processing, as you have already heard. In paragraph 5 of Article 17, there are all the information that are foreseen by the regulation to be provided in the technical dossier. So in this article, it is foreseen, first of all, that any requirements from this guidance that we are presenting today, but it should also include a description of the pre-processing step, specific quality control points for pre-processing, and a detailed specification of the pre-processed plastic input. Also the description of the post-processing and the intended use of the final material or article, and also instructions about labeling and to be provided to the converters and the final users. Now, we also need some diagrams in the technical dossier. One is a simple block diagram, okay? And the second is a more complicated piping and instrumentation diagram according to the relevant ISO, which is really an engineering diagram. It gives a lot of details of how this equipment works. Finally, foreseen a number of quality control procedures to be described in the technical dossier. For example, the operating parameters that need to be set for the decontamination process. So let's go to collection and pre-processing. I think you more or less heard what it is. Anyway, it is the gathering of the waste, including any sorting at this stage. And then pre-processing is all waste management operations following, so sorting, grinding, and so on, washing. Okay. Now, the thing that we need to remember is that the suitability of the input material will affect the final output quality. Okay, this has already been mentioned, I think, by Eugenia. So this is an important first phase. So for the collection, The requirements are listed in Article 6 of the regulation. The input comes from municipal food or retail businesses waste. As you've already heard, it has to be food contact material, so must have been manufactured according to the plastics regulation, okay, 10/2011. It's subjected to separate collection. There need to be specifications on plastic materials and articles other than PET, other materials and substances, remaining food. And finally, there must be a quality assurance system. So in the technical dossier, what is needed from the applicant, which I remind you is the developer, a confirmation that the collection is conducted according to the requirements of the regulation. And also an indication of the points of where quality control is needed. Okay. Okay, similarly, for pre-processing, we need a description of the steps, as I said before, sorting, shredding, washing, etc. In case other pre-treatment is performed in a separate, different facility, then more specific information will be needed according to the regulation. Again, we need a confirmation that pre-processing meets the requirements of the relevant article of the regulation, and also the applicant should indicate points for quality control. Okay, with regard to the specifications of the input. Okay, so as you've heard already, we need compliance with the requirement of a maximum content of non-food contact PET in the input up to 5%. Now, we need information about the flake dimensions, so range and average thickness and size distribution of these flakes. We need the range and average of the bulk density of the inputs. And we need information about the presence and the concentration of plastics other than PET, for example, PVC, polystyrene, polyamide, polyolefins, polycarbonates, and so on, okay? The presence of glues and other specifications that may be relevant to that specific process. For example, could be cellulose or metals or other things that the applicant, the process developer thinks that are important to look at in the input. Now, what we have to remember, and I think it was already mentioned, is that incomplete removal of the PET polymers during the first step, during washing, could mean that this will stay in the next step of the decontamination, where high temperatures are applied, and there is a potential for a possibility of degradation, okay, of these polymers and whatever substances are in there, to degradation to low or intermediate molecular mass products. Examples, benzene, styrene, and so on. Of course, some of these substances may be removed during decontamination because of the high temperatures as well, okay, and gas flow that may be applied or the vacuum and so on. However, it's not, this is not absolute, and also there's a possibility of formation of new substances because of the presence of this kind of plastics after the post-processing in the end. So we need to be careful in that step, and our working group looks at this point. Now, for the decontamination, I think you all heard and understood what it is. The main purpose is to remove contamination from the plastic input in order to produce a product which will be safe for the consumer using a specific decontamination technology. The only thing I need to highlight at this moment is that the decontamination process, or better, the assessment of the decontamination process, is linked to what we call the challenge test, which is a test, a study, used for the determination of the decontamination efficiency. And what we actually do is we compare the information we have in the dossier about the industrial process, and the information that is provided to us about the challenge test, because it is really important for us to accept that the challenge test, the way it was conducted, it is representative of the real life, the real industrial process. Only after this can we trust the results and say, okay, the test showed that the decontamination efficiency is that high, and therefore also the industrial process can achieve such a high decontamination. Okay, so this is a point that really needs to pay attention to. Now let's go to the equipment of the decontamination process. This is also something that we look into detail. So we need description of all decontamination unit operations and equipment, with clear identification and distinction of steps to start with, from input to output. So the decontamination process may have several steps into it. We need to clearly have them identified in the technical dossier. We need information on design and assembly of the various equipment pieces. And it really helps a lot if detailed technical drawings or photos are provided together with a text, together with a written description of the equipment. Just make a stop here to say that the more complete the dossier is, the lower the need to go back to the applicant for clarifications. And this can speed up the process. So it's for the interest of both us and the applicant to have a complete dossier. And that's why we put some effort to have this part as complete as possible in this new guidance. Now, we need to have, of course, the principal function of each piece of equipment. So, for example, we have a SSP reactor, we have an extruder, and so on. What type of reactor we're talking about? So is it a steering tank or a fluidized bed? For some special cases, so for example, special extruders, we need to know that this is an extruder, let's say, with satellite screws or planetary sections and so on. And for other type of reactors, also we need to have a good characterization, so a melt state polymerization reactor should be clearly described. Now we need the description of the heating and cooling systems, the vacuum and flowing gas, including the design and position of the ports, let's say, in an extruder, the size of the equipment, so both dimensions and the capacity, and the brand name, and if any, and the version and date of introduction of the decontamination installation in the market. Okay. Now for the size of the equipment, just a little bit more, let's be more, let's say, explanatory here. So for a conventional reactor, we would basically need the cross-section surface and the height. If there is a stirrer, the length of the stirrer, and so on. For extruders, we could need the length and diameter of the screws, the position of vacuum ports along the length of the extruder, shape and dimensions of the dies. Also for the melt state polymerization reactors, diameter and number of dies, and shape and dimensions of various metal melt tanks are used, any moving helices, blades, and so on. So the various parts of the machinery to be described in detail. We also need, as I said before, diagrams in this technical dossier. Two of those three I have already mentioned. So one is the simple block diagram with unit operations and quality control points. I mean, these are examples that we have already received, and I think the one on the right, top right part of the slide is a good one. You can see in the box that there's a description of that step. You can see what is included for each step, so steps are clearly defined, and also where we have the quality control points. The second diagram on the left bottom part of the slide is a process flow diagram, and you may be familiar with this from past EFSA recycling opinions. So this is, it has the distinct unit operations, and the schematic setup to give you an idea of what are these pieces of machinery. Okay. And finally, we have the piping and instrumentation diagram. I've mentioned it before, only to let you know that because it is such a detailed engineering diagram, it can be claimed confidential. So this is for the use of the working group and the panel to understand exactly what happens. It's not in general for the public to access it. We totally understand this. So for the quality control, we need to know what are the critical steps to be covered by quality control. So this needs to be defined and described by the applicant. And the applicant needs to think about possible consequences of failure, like in any quality assurance system, what would be the corrective actions, and what laboratory procedures may be needed to do the checks. Okay. Finally, for the characterization of the output in the technical dossier, we will need to know, of course, the type of the output. So does this process produce PET flakes? Does it produce pellets, sheets, so on? And we need to also know the intrinsic viscosity of the output. Okay. I will stop here, and I will ask my colleague Katarina to continue to explain to you more about the challenge test and the decontamination efficiency. Thank you.
Okay, thank you, Alexis. Indeed, I will continue now with the last part of the presentation. Alexis mentioned already before that there are two main concepts for the safety assessment. So we have, on one hand, the decontamination process that Alexis described, and then we have the challenge test. So how do we determine the decontamination efficiency? And this is done by the means of a challenge test that was already mentioned several times. So this is the experiment, the main study that we look at in our safety assessment. So here on this slide, we have the main steps outlined of this challenge test. So we start usually with washed and dried flakes from PET. What we saw in the applications over the last years that the input is mainly from PET bottles. Of course, other types of input can be used, but what always needs to be ensured is that the input is representative of what is then actually used in the process. So you can have flakes of a specific thickness for the challenge test, and in the process, finally, those flakes should have the same thickness or a lower thickness, so to ensure that the diffusion of potential contaminants is representative. So this is the starting point. And of course, we need to have a simulation of the contamination. As was said before, we assume that there is a general contamination of 3 milligram per kilogram PET. And in the challenge test, we need to simulate that by contamination with specific surrogates. Then we have, as a starting point to calculate the decontamination efficiency, the determination of the initial concentration. The flakes are submitted then to the critical steps of the decontamination process, and after that we also have to estimate, to measure, not to estimate, the final concentration, and based on the two determinations of concentration, we calculate the decontamination efficiency. It was also mentioned already by Constanza, the challenge test is the crucial study for our evaluation, and there is the obligation for notification of studies. So here a reminder that this study needs to be notified. So as a starting point, we look at the contamination procedure. You saw already in the presentation of Maria Rosaria that we had this list with CMOD values for specific surrogates, and these are the surrogates that we usually see used in the challenge test. So we have here a range of substances mentioned. As was said before, they represent different polarities, different molecular weight in order to cover a wide range of contaminants. So we start with volatile polar. We have usually chlorobenzene and chloroform. We have volatile non-polar toluene, non-volatile polar methyl salicylate and benzophenone, And then for the non-volatile, non-polar, we saw already an example, the phenyl cyclohexane, but also methyl stearate could be used as a surrogate there. What is important to mention here also that those surrogates, of course, they should be stable during the decontamination process so that you are actually able to measure them, of course, at the beginning, but also at the end, and you don't get like an overestimation of the decontamination efficiency. How do we contaminate the material? There are actually two modes of addition how you can contaminate or bring the surrogates into the material. The first option is that you add the surrogates to the material directly as a neat mixture, or you can use it in solution. And then you have to mix the material in order to ensure that the material is contaminated in a homogeneous manner. And afterwards, the surface contamination needs to be removed, because otherwise, if it remains on the surface, you would again overestimate the decontamination efficiency. So this is the first way how to do it. There is also the option of adding the surrogates directly to the material in an extruder, so everything is mixed together in the extruder. This already by itself allows for homogeneous distribution and contamination of the material, so there is no need afterwards for washing and rinsing. Both options, they can be done either as a contamination of the entire batch, or you use a master batch approach. So this is how you can contaminate the material. What is important for us in the technical dossier is to understand like the origin of the material, which is the type, so did you use flakes or pellets, the dimensions. Again, this is important because then we need to compare the challenge test with the recycling process in practice. The same for the size distribution, and of course we need to understand the amount, and then which chemicals were used as surrogates. In general, the contamination level that is artificially achieved by this contamination procedure should be at least 250 milligram per kilogram PET, so to allow you to measure the substance as well in the material before and after the decontamination. So once the surrogates are added to the material, there is the step of contamination over a specific time period, so the soaking and storage, and this should simulate a case of misuse. We assume in the guidance document that this misuse is for 1 year at room temperature, so the conditions that are used for the contamination, they should simulate this time period. Of course, you will not have the experiment running for 1 year at 25, so there are some conditions laid down in the regulation for migration testing, so you have time and temperature conditions specified that would simulate the time and temperature conditions in use. So you can consult this table then to also estimate the conditions that will be important for you. So it's not only migration, but also the other way around diffusion into the material. In addition to these conditions, there are conditions that could accelerate the diffusion. For example, use of solvents, so you have swelling effects, the surrogates can penetrate the PET in a faster way. So these are options that you have there, and of course there needs to be, as I said before, some handling conditions to promote the homogenization. So usually there is a mixing, or if the flakes are in barrels, they are rolled. So this is important for us to understand as well. Then after the surrogates have been soaked for this time and temperature conditions, there is the step of the washing that I said it's important in order to remove surface contamination. So we want to avoid to overestimate the decontamination efficiency. And here we need to ensure that there is an adequate removal of the surface contamination, and this will be ensured by using appropriate washing liquid, so detergents or organic solvents. No water is foreseen here for this step. In the technical dossier, again, we need to have some data or evidence to support this efficient removal of the surface contamination. And as I said before, if the contamination happens in an extruder, this is not necessary. So the washing needs to be described. We need to understand which is the liquid, which is the volume, and again the time, temperature, conditions. Then we come to, let's say, the operation or the machinery that is used for the challenge test. Alexis gave you already an overview of all the details on the reactors that we need to know for the industrial process. The same applies also for the challenge test. Because, as we said before, we need to compare the industrial process with the challenge test, and we need to ensure representativity. Also for the conditions, we need to make sure that the challenge test covers the conditions of the industrial process. And if it doesn't, there needs to be some explanation on how this affects the decontamination efficiency. Also, what we see in the dossiers that there are different scales of installation for the challenge test. So not all the challenge tests are run on an industrial process or industrial scale. They could be smaller, small scale industrial, they could be a pilot plant, a laboratory scale. So in the working group, we have discussions how these setups of the challenge test represent the actual industrial process. So what is important there is that we can do scaling up evaluation. So this is something that is also requested by the applicant, so that we again make sure that it's representative. And here I come back again to something that was mentioned by Constanza at the beginning of the session, the table of operating parameters. This is essential for us to understand, both for the challenge test and for the process, the different steps, the critical steps of the process. So the operating parameters, time, temperature, pressure, and so on. Also if it's a batch or a continuous process. We request also information on the throughput or discharge rate, depending on which mode of operation it is. And again, related to the size of the reactors, the capacity, the load, and the filling level. There are also some other types of information that are relevant. They are not usually in the table of operating parameters, but they relate to the operation. So if there is a temperature gradient, then for some specific setups, we will ask for rotational parameters, melt surface area, the degree of mixing, and also the length and thickness of pellets that are formed within the decontamination process, again to compare with what will happen in the process. What I would like to highlight is that for the challenge test, it is really important to have the precise values. We have seen in the past that there were some ranges indicated, which will not be actually the case in reality, because you do the experiment at a specific time or a specific temperature. So this, it is very important, and we will ask for clarification if it's not the case. So then, once the experiment is completed, we need to determine the surrogate levels in the initial and in the final samples. And this is done with exhaustive extraction. In order to ensure, again, representativeness and homogeneity of the results and of the samples, we need a sufficient number of samples. And this is, again, specified in the guidance document. So when we look at the samples before the decontamination, we request at least 10 samples. If there is some material transferred from one place to the other, then we would also ask for other samples, so that we really know at the beginning of the decontamination process what are the values of the surrogates. Then after the decontamination, again, we will ask for 3 samples. and each sample should at least have 1 gram. Again, what is important here that also we understand what are the sampling times and methods, so we can relate it to the actual process. And in order to calculate then the decontamination efficiency, what I said before, we look at the surrogate levels before and after the decontamination efficiency. For the applicant, it's important to provide us these calculations. We will ask that to be in the spreadsheet. And I also want to touch quickly upon cross-contamination phenomena. So this happens when you run the challenge test with a mixture of contaminated and non-contaminated material. During the process, the handling, the surrogates could move from the contaminated to the non-contaminated material. And if you measure only the initially contaminated material, you will lose this other contamination that cross moved to the other flakes. So in this case, it's important that you determine the levels in both materials. Usually they are distinguished by color, or you analyze the mixture, but making sure that the percentage, the ratio of contaminated and non-contaminated is the same. For the method of analysis, we have also some characteristics outlined in the guidance document of what has to be provided to us, because we need to evaluate the method if the results are reliable or not. So there are some information that you need to provide on the test method, detection limit, precision of the method, and recovery, so that we can evaluate the suitability of the method. And then, last but not least, a quick look at the post-processing, where we also request some information from the applicants. Again, a short description of whether there are any steps. Could also be that there is nothing, that's also fine. But then what is really important is the intended use. And here we come back to the exposure scenarios that we saw before. We need to understand which is the intended use to agree or to evaluate properly the scenarios. So that is specified in Appendix C of the guidance. These intended food applications that have to be indicated in the dossier. Also, like time and temperature for the final use, and if there is something that is not suitable. In the case of mechanical PET, we already heard that it's not suitable for microwave and oven use in the technology as defined in the regulation. And this is my last slide. The last section of the guidance is the self-evaluation of the recycling process. So we heard already by Eugenia and by Maria Rosaria that we have these two concepts, the residual contamination and the modeled contamination. We will request from the applicants to do a comparison of those two values and to evaluate basically whether the use that is requested fits with the criteria and with the results of the challenge test. So in Appendix D of the guidance, we have already calculated the C-mode values for the most commonly surrogates that we usually see in the challenge test. So you can consult this guidance document, this appendix, and then compare your results with what we indicate there. Again, if the CRES is not in line with the C-mode, you already saw before what to do. You need basically to dilute the recycled PET with virgin PET. So this was the example that Maria Rosaria gave earlier in the presentation. So with this, I will wrap it up here for the content of the technical dossier. I will give the floor back to Sandra. Thank you.
Thank you very much, Caterina and Alexis, for presenting the content of the dossier. And with this, we finish with our presentation, and we open the next session, which is a discussion with the panelists. Before that, as I said at the beginning of this meeting, we would like to address the questions that were received during the registration, because this will have priority above all the other questions. We received 16 questions, which were falling under the different remit of either Commission or EFSA. You can see the topic over there. We tried as much as possible to address those questions with our presentation. Still, some of the questions still need to be addressed. These are some of the EFSA's related questions dedicated to the application procedure, and I think Costanza addressed all of them in her presentation. In any case, if you have any further doubt, you can contact her directly or just check the corner. There are some questions that were in the EFSA remit regarding scientific issue. One was related, was a registrant that commented on the during the public consultation when the draft document of the guidance was submitted for public consultation, and wanted to know basically what happened with the comments. As Catarina said earlier, all the comments that were received and all the answers that were given to the comments are in an annex that you can consult publicly, which is attached to the guidance. Just for your information, there were some comments on the migration criteria and on the contamination of final article produced by recycled PT that were taken into consideration and caused some changes into the guidance document. Specifically, for example, benzene, there was a part on benzene that was removed because the experts decided that the scientific evidence was not supporting enough sufficiently for the moment, including that paragraph. We have then another question that I will read out loud, which will be the guidelines to follow according to Regulation 2022-1616 to calculate the percentage of recycled material inside the product sold on the market. I think maybe Maria Rosaria is going to answer this question.
It's working? Yes, thanks. Thank you. I think that we already addressed this kind of question, but there is not a guidance, written guidance, but it's a self-regulating because you know your CRS and the C modes are tabulated, so you can easily compare and calculate how much dilution you need to comply with the C mode. Thank you. I think it's enough.
Okay. I hope it answered the question. Then we have another one. Okay, the second one is the new EFSA needs are requesting measurement down to 0.00015 milligram kilogram food simulant. In the recycling plant, I can measure material directly. Migration is done in a bottle, not allowing me to do inline inspection. Most labs are simply not able to measure as low as that amount. Maybe, Eugenia, you can address this.
Thank you. It's working, yeah. Okay, thank you for the questions. I think that this statement is wrong statement, it's not correct, because EFSA does not require for the laboratories to measure at this very, very low level, below 1 ppb measurements. This is the migration is below 1 ppb, but this migration, what is requested by EFSA is to perform a challenge test and to determine the quantity by extraction inside the PET material which correspond to this migration. So the laboratories do not have to perform migrations with the bottles and measure the migration at this level of ppb, of below ppb. The measurements should be performed by extraction before and after the decontamination process in the challenge test. So in these cases, the level of measurements at the below 1 ppm and around above 1 ppm. So this very, very well established methods exist in all laboratories and validated methods.
Thank you, Eugenia. I think this clarifies this issue. We go then on with the questions on the European Commission remit that were addressed during Bastian's presentation. These were. And then we have a few more that were not and were deliberately left out, and Bastian maybe can address them now. So, how can the 5% non-food plastic input rule be reinvented to accommodate sources of materials such as separate collection schemes?
Well, the 5% rule is laid down for the present technology of mechanical rPET recycling. That 5% rule, which is there, cannot be reinvented. But of course, there can be a novel technology development which uses a different kind of input, because input is part of the technology, using, for instance, a much higher percentage of non-food contact plastic, which will then probably require a re-evaluation sort of the background contamination level, and then that can be evaluated in the end by EFSA to see whether that technology will be suitable and what sort of higher decontamination requirements would be required from that technology. So yes, it can be well, reinvented, if that's the right word, via the novel technology procedure.
Yes, thank you. So yes, with new technology this can be achieved, not currently. The recycling facility must conform with 5% maximum in non-food plastic input. In a first, recycled produces non-decontaminated PET flakes is not concerned, but the following recycler to decontaminate flakes and transform it into pellet is affected. Is that correct?
Well, no, not really, actually. First of all, in the terminology of the regulation, the real recycler, so what we would call the recycler, is actually the operator which carries out the decontamination. So there would not be a first and a second recycler. It would be the following recycler, which is actually the recycler, which means that the recycler needs to use an input which conforms with the regulation. Of course, it's impossible to check on the basis of flakes whether it contains this 5% maximum, because how would you know from flakes? So basically what needs to happen is that what is now addressed as the first recycler, who is apparently shredding and probably pre-washing the flakes, will then need to ensure that this 5% maximum is there, and that he will need to certify so that the second, as I said, the recycler is able to just use that certification to comply with the 5%. So the first would indeed be affected by it, because he needs to carry out this separation and certify that he does properly.
Okay, thank you. I think this also clarifies. Availability of case study to understand complexity of actors concerned or not within the recycling chain?
Well, I struggled a little bit with understanding the question, but I assume it's about the different configuration of actors that can work. In fact, the previous question is a little bit an example of that. I think what we instead did is we tried to write the regulation in a way that it is general. It covers all cases, also by making clear that there is pre-processing, there is the decontamination, and that there is post-processing. And normally different configurations of actors, which may indeed exist, should fit in there, or they do the pre-processing, or there is the sort of single decontamination step, or they are in the post-processing. So that is how it works, and I don't think we need a further case study, and that case study is unlikely to cover necessarily all configurations of actors which may be operating in the supply chain, and I don't think that there's any reason also to make a difference. We took care of sort of covering, by means of the structure of the regulation, all the requirements for these different actors, and so I don't see that there is a need for such a case study.
Okay, thank you. I don't know if the registrant who sent this question is here, and if the question was understood correctly. Otherwise, okay, seems it's okay. Okay. Any specific condition on regulatory discussion going on regarding biodegradable FCMs?
Well, not in this framework, really. So we're talking here about plastics recycling, and basically to biodegradable FCMs, two pieces of legislation would be applicable, which is Regulation 10/2011. So biodegradable FCM would first of all need to comply with Regulation 10/2011. Then on present, of course, since RPET is not biodegradable, there is no suitable technology for recycling such plastics. Well, I don't know exactly what is all biodegradable, but let's say PLA is biodegradable. Then you'd need to have a suitable technology for recycling PLA in this context. Of course, there are several discussions ongoing about biodegradable FCMs at different levels. For instance, under environmental legislation, it's relevant, but that isn't concerning this particular discussion.
Okay, thank you. Okay, we have some questions on novel technology. I don't know, maybe we can answer that here since they were, I think, skipped during the presentation. How to speed up the process to allow novel technologies and food grade for polymers other than PET?
Yeah, so the novel technology is defined under the regulation. So basically, what it requires in practice is that there's a data gathering phase of 2 years, and then there will be an evaluation, and that we will follow as far as possible. We're at the moment working on an inventory of novel technologies because the registrations we got were a bit confusing sometimes. We got too many, and some seem to be rather dealing with recycling installations rather than the novel technology itself. We're currently sorting that out. But there will be a certain moment, and that moment is actually approaching rather fast, where we will start asking EFSA to do the first evaluations of novel technologies. In particular, novel technologies which are big on the market, we will probably prioritize. Also, novel technologies where we think, the data is really now solidly available. Then that will go ahead. But yes, we will be using the novel technology procedure as it's laid down in the regulation.
Okay, I think there are some uncertainty around the validation that you have partly addressed already, but if you want to add anything regarding this uncertainty that this question...
Yeah, well, I think regarding this uncertainty, I think we should all keep in mind that the whole novel technology thing is new to everyone, also to us, but to many applicants. So what we will be seeing, I think, in that first phase, which we also just addressed, we will see that we will really start to define how to handle that kind of data, and there will be a bit of a learning phase, unfortunately. I think there are different levels of data interpretation. So we at the Commission will be interpreting the data to a certain extent, but the real work will need to be done by EFSA, and I think also EFSA will need to build as soon as they are confronted with that data on how to interpret it, and that might actually be different per technology. So there we will have to learn. What we will see is that perhaps some of the data may not be sufficiently covering the information, and I think therefore it's very important that people think, perhaps based on the present EFSA evaluations, also on the guidance which we are discussing today for recycled PET, what sort of data could be needed, both on the input material to characterize the input, but also on the functioning of the technology itself. So I think therefore this guidance is also very important to novel technologies to see, is it wishful thinking what we are doing on our novel technology, taking into account how actually the RPET is being evaluated? And I think that is a very important aspect. And there maybe also EFSA may say a few words, because this is more on the remit of EFSA than in fact on our remit. Then I'll take the last question, actually. Any changes in the list of specific NIAS? Any updates on standards to measure NIAS? Well, on the first list of specific NIAS, I've never seen a list of specific NIAS in the regulation or anywhere actually. So this is non-intentionally added substances. So what the regulation actually asks, because we want to have a good overview of what sort of substances can be present both in the input and the output of novel technologies, is to do more or less I would call it, the regulation doesn't say that literally, but a non-targeted screening of these substances. I know that quite a few business operators are working on that. And then the regulation asks to identify the 20 most frequently occurring substances. For instance, in RPET, a typical example would be acetaldehyde, which everyone knows it is occurring all the time because it's related to the PET itself. So that would be on that list probably if it were for RPET, but that might be different for other substances. But that is to be identified by the developers of the novel technologies, because they are working on it. They see the plastic, they see the data, and that is what needs to be done. So we will not be updating that list because it doesn't really exist. And then the standards to measure NIAS. Well, what the regulation says is that in a couple of days' time there needs to be a proficiency test done. Well, whether people will be able to meet that is a little bit questionable, but I think what is the important thing is that we see quite a few business operators who are actually working on understanding how to properly measure NIAS, and there we will also learn, and maybe we over quite a few years we will be moving towards good standard methods which can be used for this purpose. And unfortunately we couldn't refer to really ready-made methods at the time we put down the legislation. So we're learning now, and we probably will need to evolve over a while on these kind of methods. So I hope that answers it.
Thank you very much, Bastiaan. I wanted to add something regarding the novel technology, since I think, Bastiaan, you made a very good point. We really wanted to have this info session to get you to understand what we really prioritize in our evaluation, what is the rationale behind, and this can indeed be useful for when you plan novel technology. I mean, it's not going to be the same, but at least I hope you had a taste of what we really focus on. And please use this information also for developing new technology, and then we will have a faster job, I think, and more efficient. I think we finished with the questions that were submitted during the registration, so I would open now the floor to the audience. If there is anybody who want to ask any question, we won't answer questions on specific cases because these are dealt with in a different way. But if you have any general question, please. I think there is 3 that I see over there, here, there, and here. Please introduce yourself before, and then we can see.
My name is Casper van den Dungen. I represent plastic recyclers. I have a very simple question, because I was very surprised by the last presentation that you said it's about the surface cleaning. And you said very intentionally not use water. So if you want to do it with the suggested liquid you are trying to use, whatever detergent it is, then you are introducing that substance as another contaminant, because you will not clean the surface only with that. You will leave then detergents on top of it. I don't understand that you say not use water to surface clean the contaminated flakes.
Please, Eugenia.
Thank you for the question. Yes, yes. For after the artificial contamination of the flakes with surrogates, we know that there is. We know from the data we had up to now that there is surface contamination. This surface contamination shall be removed before going inside, because this surface contamination does not represent the actual contamination of PET. So this should be removed, and water is not capable to remove all these organic surrogates used. So there is a rinsing. The applicant may perform washing using, for example, detergents or alkali, or rinsing with, for example, 10% ethanol. And in any case, this is an artificial test. This is a challenge test. And we measure, the applicant should measure only the surrogates used for the contamination. The decontamination efficiency determination is based only on the surrogates used before and after the process. This is so. There's no any problem with any solvent used for rinsing.
Maybe to verify, maybe I didn't express or it came across a little bit different. It was stated as you cannot use water at all. Maybe that's the interpretation that said if you use water, including detergent, etc., then it's fine. Then you clarify the point, because now it was stated no water. And then I said, okay.
No, only water. Because we have seen cases where the applicants...
Then it's fine. Then it's a misunderstanding in the wording. Okay, thanks.
Okay, thank you.
Okay, I'm glad it's clarified. Next question was here.
Hello, my name is Lisa. I'm here because I've made already a lot of applications to EFSA, especially for scaling of processes. And my question is, can you please confirm that this new criteria does not affect positive opinions which are already published? I mean, you write in the guidance that it's applicable to only initial applications and modifications. So I think it's clear that all the old
I can take this. I mean, we are applying the criteria, the scientific criteria, to the application that are incoming. Now, a guidance document is not legally binding. So, of course, you might say we had an old guidance and now the new guidance has been published in June 2024, but the scientific development and knowledge and the regulation were already out there. So our expert couldn't ignore what happened. So the current application are being evaluated according to all the criteria that our experts consider valuable. For the opinion that are already published, they are already published.
Thank you.
Yes, and we are authorizing on the basis of those opinions.
Thank you.
Oh, thank you. It's Jez Blake from Bury. I don't want to linger on it, but I just wanted to just dive back into your last comment, Bastian, about the screening for novel technologies. I know we're not here for that, but in the discussions about the general screening of materials for a novel technologies assessment, I think the initial description was to look at compounds up to 1,000 Daltons in molecular weight. Now, obviously, going above the GC range is much more complicated because it involves LC and other sort of methods. Is there any flexibility in the approach if the application goes in and it doesn't do it batch by batch on things that are outside the GC range?
I think at the time we verified what could be done by GC and what could not be done by GC. I think there were a couple of publications out of laboratories which were sort of working in that range. But I think in general there is no method, as I said, laid down at the moment. So I think there's definitely going to be flexibility because first of all we have to see what sort of method really in the detail will be originating from all this work. And in the end it will need to be EFSA who does the evaluations, who will be looking at does this give us sufficient data or not. But of course, the work now we're doing on the basis of what can be done and then the requirements of the regulation also. So we have to see and how strong this 1,000 Dalton criteria in practice comes along. But of course, there are methods which can do so, and also substances up to 1,000 Dalton could be problematic for toxicological reasons. So I think that is what was at the time considered.
Okay, thank you.
I think we have another question. I saw somebody. Okay. Okay, Anna?
Thank you for this very interesting morning session. Everything was very useful. I'm Anna Gergely from my own company, Cambridge Consulting. Just a very small clarification. We are always talking about this 5% tolerance of non-food use, PET. Do we mean PET which was initially not intended for food use, hence it didn't need to meet the union list restriction, or PET, which was potentially suitable for food contact but was used in detergent or cosmetics application, because it's not very clear. And the examples which I think one of you listed, it really shows contamination in the non-food type of use, hence cosmetics, but I don't know why does it matter, because you would remove them anyway if your surrogates cover the whole spectrum. So I always thought that this 5% meant in something which was not intentionally but didn't meet the union list.
Any volunteer?
Should I then?
Yeah.
perhaps?
Yes.
So let's take a look at what the regulation really says, and now I hope I know it by mind good enough. So we're talking about Article 6. So Article 6 requires compliance of plastic which is being recycled with 10/2011. It also requires separate collection, and in that light, I think it should be seen. So this 5%, which was initially coming forward from the EFSA guidance, was then taken on into the regulation. And in my view, indeed, it could contain plastics which are not necessarily compliant with Regulation 10/2011, because the 5% is more or less seen as a kind of contamination. However, I think in the PET sector, it has been demonstrated several times, and it is being demonstrated, that actually all the PET which really gets collected by means of the separate collection system, which would be accepted for such input, would actually comply in practice with 10/2011, simply because in PET the level of additives is very low. There are no strange additives being used, and therefore we will not have that problem. But I don't think it will be possible to exclude the potential presence of non-compliant PET in there, and I think that's also one of the reasons why it's being limited. So that indeed, if there would be some strange additive present in the end, its risk will be limited. And I don't think that the challenge test or actually the decontamination process necessarily guarantees that such additives are taken out, because they could be of a very specific nature. So I think therefore this 5% is also really quite important.
Yes, and that would be my understanding, but that it has nothing to do with what type of potential chemicals might migrate and contaminate PET in non-food use, like from a classical, I don't remember the examples you use from cosmetics. Because it would be removed anyway, potentially, if it is the surrogate represents it.
Maybe Eugenia wanted to clarify.
Yes, just to add something, that the contamination efficiency, as I said, measures the ability of the decontamination process in order to remove every contaminant irrespective of the source. So in this case, we check all of this. Yeah.
Okay, thank you. Any other question? Okay, ladies first.
Hello.
Hello.
My name is Laura Probst. I'm from EREMA. I have a question regarding the audit. So as far as I understand the talk today and the regulation, it is compulsory for every recycler and recycling facility to get audited. I'm in contact with some of our customers, and especially outside of Europe, it's quite hard to find a national authority or some kind of governmental body. So my question is it possible that if a neighbor country audits the recycling facility? And another question, I think it will be quite hard and also frustrating for our customers. They already have an authorized process, they have a recycling installation, and they are not allowed to sell the rPET on the European market because of a missing audit. Thanks.
I think, Bastian, that's yours.
Yes, it's definitely mine. Yeah, we are in contact with quite a few authorities across the world, literally, who can or cannot do this kind of work. And what we will see in the end is that there will be certain countries who will not be able to perform such audits, which means that although we're not making a great speed at the moment, because we're really giving people the time to establish it, it means at a certain moment that we will start suspending recycling processes, whether in the EU or whether outside of the EU, which have not been properly audited. So that will happen. So yeah, the unfortunate truth will be that in some countries there will not be competent authorities who can do that work and who will then not be able to export recycled PET to the EU. Otherwise, they can do whatever they want with that recycled PET, but they can't export it to the EU. That's then the consequence. Yeah, this is how this will develop. And yeah, you had a further question?
Is it possible that the neighbor country?
Yeah, oh yeah,
does
sorry.
the competent other
Yeah, so no, that's actually not possible. Because basically the regulation says it needs to be the competent authority on the territory where it's located. The reason that we are actually requiring that is that we want to have independent oversight over the production by a certain producer. And then we are unable to set out different rules than we would be applying in the EU to such oversight. And in this case, yeah, it's the audits, which are the oversight by a competent authority in the territory where it's located. And therefore, yeah, another competent authority simply doesn't have any legal jurisdiction outside of its territory where he's competent. In fact, in some member states that can even be locally, another authority can't start, even in a city next door, can't start doing that kind of work. So that is basically impossible. So it needs to be someone who is really responsible legally from a country who can do that kind of oversight.
Okay. Yes, we have another question here.
Oh, thank you. In the regulations about PET, it discusses that the PET is collected for recycling. We've certainly had lots of inquiries in the past couple of years about lots of places that are looking to recover bottles from, ERF, energy recovery facility inputs, or even landfill inputs. So if bottles are recovered, they're not specifically collected for recycling, and this has been a bit of a grey area about would they still be able to be put through a food contact process or not, just because they weren't initially collected for recycling, but subsequently then recovered for recycling?
Yeah, so I think the requirement in Article 6 is really that they are separately recycled, or separately collected, sorry. So the collection has to be done separately. And the reason for that is that if it's not done separately, they will be collected as part of the residual waste, and there is no way to have a proper definition of what would be the content of that residual waste. So there needs to be some form of separate collection which sets out rules for the specific materials which can be entered into that stream. I don't think that any landfill will have been operated using such rules. So basically, you can't see collecting from a landfill as separate collection, because it's just basically residual waste which has been left to smother for a couple of years. And I think also what's not possible, and I think that is a more realistic example, at least I hope, is collection of residual waste and then having a facility thereafter which separates out that. That is exactly what we do not want, because there you have the issue. So what went in together, and what sort of contamination can we then really expect from there? So the separate collection has to be done at the point of collection, which means it has to come from separate bins, essentially.
Okay, potentially the second example, which I think is more common these days, would that also exclude it from a separate novel technologies application in relation to that?
No. So novel technologies are able to sort of bypass anything which is in Article 6. That is what is also laid down in the novel technology procedure. Of course, there needs to be a good argument for doing so, and there needs to be proof that that then can be done safely. So indeed, through a novel technology, you would be able to say, well, we just collect everything together from residual waste, but our novel technology cleans a lot better, so it can take out different plastics and additives from these plastics, for instance, because they could be there together, it has a good sorting, etc. And then, yes, it would be possible if the novel technology is able then to in the end, demonstrate it properly.
Great, thank you.
Okay, I think one last question.
I'm from Aliplast company, and I have a question on food contact materials or feedstock to be used in food contact material coming from outside EU. We discussed today quite a lot about the fact that one of the pillars is the fact that the feedstock should come from 10/2011 compliant material, but 10/2011 is just operating in Europe. So how, if you use feedstock which is coming from outside EU, how can you prove that this is 10/2011 compliant? And in case there is a way of doing that, how authority controls can check this?
Yeah. So I think what is very important is that we want to establish at a certain moment a full certification of the input material, which means from the point of collection through the sorting until it is actually used as input by what we call a recycler. That certification should ensure that that material is compliant, that there are good procedures being used by the companies, that they operate under good manufacturing practices, etc. Now, then the question is indeed, so how to deal with this requirement of 10/2011, which, may be a bit hard to meet if you're outside of the EU. Well, again, for RPET, the case can be made, and that is probably what we're also going to accept, that people just say, well, actually, the PET which is being placed here on the market is maybe not officially compliant with Regulation 10/2011, but it is fully similar because it doesn't contain any of these additives, and there can be verifications, for instance, with locally operating producers of packaging material, etc., to see whether that is actually the case, and in many cases that will be confirmed. And we have seen that before, and I think even in EFSA's work, I remember initially that kind of proof was being given, but I don't remember it exactly at this moment. But yeah, that is how we're looking to work. But given that PET might be a bit of a special situation here, indeed, because of the aforementioned sort of low presence of additives in PET, etc., it might be much more difficult for all other plastics. That is what I would be expecting. And this equivalent might not be so easy to establish, which means that, essentially that kind of recycling streams would become blocked because probably it couldn't be proven unless, again, and there we go also to the previous answer, the novel technology would be cleaning so good that it can say, well, actually, all these sort of potential non-compliant additives, etc., would anyway be removed by the technology, and then of course it's fine again, and because then also the 10/2011 provision in Article 6 could be sort of overridden by the novel technology. But as I gave also in my previous answer, there needs to be really solid proof at that moment, which is also accepted by EFSA then at that moment, that would be the case.
Yes, but in any case, I would say the importer of such these materials should do, even in case of PET, some additional tests in order to prove that there's not other, I would say, additives or potential contamination which are coming from not being compliant with 10/2011 in case. And in that case, how this control authority should check this?
Well, I think there's no real role for the control authority other than to check the certification which has been done. So it's not the control authority which will be checking this, it is the certifier who will be checking this. And therefore, also at a certain moment, there will be third-party certification required. And we are thinking of indeed having actual certification of the product, which is presently not required. So there's no actual certification at the moment of the plastic input, and that might actually be required for this purpose.
Okay, I think that was the last question. Otherwise, we run out of time. Thank you very much for your questions. In case we have not managed to answer all your questions, or in case you didn't have the time to ask what you wanted, please contact us now during the lunch or via Ask a Question, and you will find the specific link in the presentation that will be available in the EFSA webpage once in a few days after this info session is finished. You have.
Yeah, I have just one small addition to make. Okay. I see in the back of the room there's my colleague Mario Nachtzaam. He's responsible actually
hello.
nowadays for the plastics regulation, so Regulation 10/2011. He's also responsible for the quality amendment. I just shortly wanted to introduce him. If you want to take the opportunity to have a chat with him, maybe that's an opportunity now. Or maybe you just run away before you get a chance because he's anyway in the back. But yes, so it will be good to know. And also here in the front is, of course, but I think you already knew him as Apostolos, who is dealing with the registers, who is dealing with the authorisations. So these are the two team members who could be really quite important to you. I think Apostolos, in case of recycling, obviously the most. But if you are talking about compliance with Regulation 10/2011, it's Mario really. So yeah, that was the short introduction I wanted to do.
Okay, so now you know where we are. We also have two of our experts in the front row that are part of the recycling working group, so you're also welcome to talk to them here in the first row. As I said, the recording will be available in EFSA as well as the presentation, and there will be an evaluation survey that you can fill, and it will be sent to you shortly after the meeting. To wrap up, as I said before, the intention of today's info session was to make you aware of the way we work and what we really give priority to. And I hope it was clear, the message was clear, and I hope we can interact with you now during the lunch that will be somewhere upstairs. Somebody will take us, I suppose. Okay, and then I'm looking forward to seeing you again for the novel technologies guidance that we will develop in the future. Thank you very much, and thank you to all the speakers.